Welcome. We talk about the genes that played a significant role in WHO classification 2016 of the CNS tumors, and we will discuss 7 of them and the idea behind them, why what's the clinical implication of these genes. We're starting with the IDH1 and IDH2. This is the isocitrate dehydrogenase enzyme. So this gene, in normal cells, the isocitrate dehydrogenase enzyme acts on isocitrate and creating alpha-ketoglutarate, which is one of the steps in tricarboxylic acid cycle, the Krebs cycle for cellular respiration and ATP production. But in abnormal cells, in tumoral cells with mutation in IDH1 or IDH2, the muted enzyme will act on alpha-ketoglutarate and creating what we called 2-hydroxyglutarate, which is a small oncometabolite which promote glycogenesis and associated with low-grade astrocytoma, oligodendroglioma 1 and 2 and 3, and also with a secondary glioblastoma with better overall survival. So it's a good mutation. It's a good prognosis if we find that the glioblastoma is IDH mutant. Comparison and opposite to if we didn't find the mutations, which called the IDH wild type. Other gene is the O6-methylguanine DNA methyltransferase. So basically, in the normal cells, DNA sometimes having the methylation, so mean that there's some methyl group attached to the one of the bases of DNA, usually CG. And if usually the cell had what we call effective O-methylguanine DNA methyltransferase enzyme, will remove this methyl group from DNA and repair the DNA instead of become broken and leading to apoptosis. This enzyme is functioning well in the normal cells, so we'll have a DNA repair. In the tumoral cells, the enzyme is defective. This gene is defective, so the enzyme is defective. So here temozolomide will come to action. He will alkylating agents, or that means he attach alkali group to DNA bases. And because of the tumor cells doesn't have a good effective enzyme that can repair this DNA, that will lead the cells, the tumor cells, to be destroyed irreversibly and apoptosis. So this is a good mutation and will lead to good response to temozolomide. Thank you.